Hi everyone, welcome to our STEM News broadcast for week 7 of spring quarter of 2022. We are STEM News Nexus at UCLA, blog of preprints and new publications. So let's get started. I just looked over this preprint on modeling human blood-brain barrier. It's entitled "The Three-Dimensional Microenvironment Regulates Gene Expression, Function, and Tight Junction Dynamics of iPSC-derived blood-brain barrier microvessels" by Linville et al. And I will post all this information in the description. Just email STEMNewsNexus at ucla.edu for any accessibility related inquiries. And I'm referencing the version posted on May 5th of 2022 and citing specifically pages 1 through 3. So essentially the blood-brain barrier is made up of specialized brain microvascular endothelial cells that filter elements entering the brain from the blood. It's difficult to model this tissue in vitro using stem cells because the cells cultures lack certain phenotypic features, for example, maintaining a resistance of 1,500 to 8,000 ohms, and also lacking a certain set of gene expression markers. So these researchers asked if the lack of biomarkers is because of the mechanical differences around the induced brain microvascular endothelial cells. If yes, then induced brain microvascular endothelial cells and induced pericyte-like cells will have more phenotypic character in their 3D instead of 2D structures in vitro. So they characterized how the 3D structure augments the BBB phenotype by first looking at how the structure looks like, because they'll kind of grow these IBM-ECs and cultures and then start structuring them into 2D confluent petri dishes or 3D gel print structures. And then they'll show how the tight junctions behave during injury and homeostasis. They applied 3 different perturbations and assessed it. And they also did some transcriptomics on these same cells in these different microenvironments. And then they just conclude that the 3D environment affected the behavior of the cells in culture. And yes, so I totally am interested in this preprint. It's relevant to what I'm researching. So I think that this is really cool and I'll be reading more into it. I hope that everyone has a great week 7. And yeah, okay.